As we come to learn more about how the DNA in our genes both ties us to and differentiates us from each other and all living things, we are approaching a point in technological history where a world like Gattaca, a place where our genes determine our worth, is not only possible, but possibly imminent. How did we get here? How did we come to know what we know about genes?
The first breakthrough that led to genetic engineering came in the 1950s by an American and a British scientist, Watson and Crick. They worked out how the genetic code works. And it's a chemical called DNA.
Watson and Crick established the actual physical structure of this DNA. And what they showed that was so critical is, firstly, the DNA is a very large chemical molecule. It's made up of small beads strung together, and there are 4 different sorts of beads, and the order in which they're strung together is the basic code.
These beads are actually the molecules: adenine, cytosine, guanine, and thymine, or GTC and A. As Watson and Crick were studying the arrangement of these molecules, they noticed that G was always coupled with C, and that A was always with T. In addition, they noticed that by connecting these molecules by their hydrogen elements, the same shape was created, and by stacking and twisting these pairs, they discovered the double helix structure.
Because the way in which they sit next to each other is absolutely rigidly determined by their chemistry, if you pull them apart, each one makes an identical replica of itself.
The great mystery of genetics was suddenly dissipated, and it was straightforward chemistry.
The second breakthrough came in the 1970s with a British scientist called Fred Sanger. He worked out not how the code works, but how you can read it in a practical way.
In order to find a specific gene within the DNA code, scientists had to find the order of the GTC and A molecules in the DNA strand. Fred Sanger used 4 different chemicals that would attach to the GTC or A molecules in a fraction of a DNA virus. By doing this, one of the chemicals could locate all the G's in the DNA strand. Another would locate the T's, another the C's, and then finally the A's. This process, developed by Sanger, is known as gene sequencing.
Over a 20-year period, we went from having the Watson and Crick paper to having recombinant DNA methodologies. That was of monumental importance and generated companies and generated enormous amounts of new knowledge and gave us an ease with the handling of genetics that we had never had before.
At this point, scientists understood what genes were and how they worked, and were beginning to understand how they could be manipulated. However, in order to find a specific human gene, they needed to determine the sequence of the GTC and A molecules that are specific to the human species. This sequence is the human genome, and the initiative to discover the sequence is called the Human Genome Project.
As a logistic exercise, it was absolutely huge. They were putting 73 million letters of DNA onto the web free every night, just in order to stop it being, in a sense, grabbed by the commercial sector.
Mapping the human genome is a fantastic achievement. I don't think it's necessarily in itself going to lead automatically to miracle cures or to crops that grow anywhere. The code has been read for a human being, but then you've got to apply it.
It gave us a catalog, and it gave us, it's not just a catalog in the sense that it's a set of different objects all put together. It's also this long string of information that is seen as having various forms of codes in it that tell us different things, that tell us not only the structure of proteins, but the structure of inheritance, the structure of development, the structure of gene control. Many different things are buried away in the structure of DNA.
Having a map of the human genome would open up endless opportunities. But what scientists were most excited about was how this could aid in the search for cures of genetic diseases. However, as benevolent as this intention is, some of the new technologies in gene therapy have become quite controversial.
The cells that people get excited about, very excited about, are the cells that are present normally only in very early embryos, and they can give rise to lots and lots of different tissues. If you could get those cells, you would be able to grow a piece of pancreas that would help a diabetic, or a bit of brain that would help someone whose brain was deteriorating because of Alzheimer's.
It sounds amazing, but actually it's not that much different to what we already do routinely in terms of bone marrow transplants, where we can collect bone marrow and give it to someone with the same tissue type, or even collect your own bone marrow, treat you for a disease, and then put your bone marrow back in again. In the future, I think we'll see it being used for repairing livers and skin and broken backs, for example, where you could repair a broken spine because our body can't do it for itself. But in order to get into stem cells, we have to start working with another scary word, cloning.
Dolly was born in July 5th, 1996, but it was not because people were interested in human cloning. They were interested in pharmaceuticals, the idea of modifying the genetics of a sheep udder, and so that pharmaceuticals could be produced inexpensively by sheep and extracted from milk. So we're talking about pharmaceutical production. They didn't even think that it would create the furor that it did.
The sheep was the perfect slogan animal for this radical new technology. People were taken aback that you could actually make an identical animal out of the DNA from its, what do you call it? Parent? No. Grandparent? No. Progenitor? We love the idea of this plump little sheep that's a copy of something else. I think in a way it's the only sort of safe and happy feeling we have about cloning.
The word cloning originally simply meant making copies of things. It was a routine process in the laboratory. But then people realized with Dolly the sheep that you could actually clone whole human beings. In theory. In practice, we're not going to, because it would be profoundly dangerous to try. Dolly produced hundreds of profoundly disabled offspring before they got one right. So we're not going to go in for what's called reproductive cloning. But what we are going to go in for is therapeutic cloning. So if I had cirrhosis in the future, instead of waiting for someone to get run over so we could ask for the liver, what we might do is take a nucleus from one of my cells, put it into an embryonic cell, persuade that cell to turn into a liver cell, grow a large quantity of it, and then effectively make me a new liver.
Now, a lot of people believe that when something has the therapeutic potential and the potential for understanding that cloning and stem cells provide us with, which is enormous, that we have to look very carefully at our moral precepts. Well, isn't there a difference between a blob of cells and a person?
One day, in principle, when we understand all this, we'll be able to use synthetic chemicals and do it all in a test tube. Until then, the only access to these cells that can turn into pretty well everything are either very, very early embryos, which haven't turned into anything yet, so still have the potential to make all tissues, or else this trick of reprogramming, which was discovered with Dolly the sheep, which needs egg cells in order to set it off.
We don't know how to do these things today, but we'll never find out how to do them unless we work on them. So we're in a sort of catch-22. You have to be able to do the research, but the people who are trying to block the research are saying, well, since it's not around the corner, we can block the research. And it's not around the corner because the research is blocked. There we are.
In Gattaca, for the first time, we're asked to examine an age-old myth in a new light. What will happen if we can suddenly control the basic essence of life?
Controlling our own evolution in the future has become technically possible. It's not something that we're easily going to do, but you can see a short distance down the road these questions being asked.
Genetic engineering began 200 years ago and has been accelerating ever since.
People are fascinated by the notion that we could somehow engineer ourselves. And that's because it fits into this metaphor of here we have reached out and we have controlled the natural world around us. We have in fact fashioned the human environment out of our own will. The new technologies that are emerging from our understandings of our own biology are very troubling to lots of people. And there are a variety of reasons that are put forth as to why that is so. Foremost among them is that we are somehow playing God.
Many people look at genetic engineering and say that God made us in a certain way and we should accept what we get. But in fact, people don't realize that we've been rejecting what we've been given at birth for over 100 years with the use of traditional, what is now traditional, medical technology.
The big question in genetic technology is who gets to decide? Will the government say you can't choose your child on the basis of intelligence? You can't choose to make a child that won't be a criminal. You don't have to have a big imagination to see where that's going. People want to know now, can the state say you can't have a baby? Can the state say you can't engineer a baby? Who decides? That's the big question.
Of course, it's illegal to discriminate. Genoism, it's called. If you refuse to disclose, they can always take a sample from a door handle or a handshake, even the saliva on your application form.
Genoism is a term that was dreamt up to explain a phenomenon of the future. The idea is that when people start to make choices about what kind of family they want to have, what kind of kid is good enough, that fashions emerge so that people begin to think of a certain kind of child as being good enough, and it spreads. Genoism emerges when you don't have the right kid, when your child is a product of luck, as in Gattaca, and that luck suddenly becomes something we demonize.
Neurological condition, 60% probability. Manic depression, 42% probability. Attention deficit disorder, 89% probability.
I teach about Gattaca in my class, and I ask them whether they think that genetic discrimination already occurs. And I tell them that there actually is a chromosome right now where if you have this chromosome, you pay $500 more in auto insurance in New Jersey. And of course, the chromosome is the Y chromosome.
The use of genetic profiling is something that we simply have to face because the opportunity is there. I think what you have to do is to say, we need a form of universal health care that doesn't ask the question, what is your genetic determination? And so we've got to look on the political side at the implications of what the science is telling us. So this is a very real issue right now.
I think the next century is going to challenge our vision of ourselves as no previous century has. The real issue is whether we can continue to face the possibilities of the future or whether we're going to pull back in fear and turn over that exploration to other braver peoples because, in fact, the next frontier is not space, it's our own selves.